Good afternoon. My name is Olga Anczukow, and I'm an associate professor at the Jackson Laboratory. My lab is interested in alternative RNA splicing, which is a process that generates from a single gene multiple mature mRNA isoforms and potentially multiple protein isoform, which can have similar or quite distinct function. This process is frequently altered in human tumors, and we have known for many years that tumors often exhibit distinct isoforms compared to corresponding normal tissue. In this example, the middle exon is included in normal tissue and becomes skipped in tumors. These aberrant RNA isoforms are found in many cancer-related genes and impact cell proliferation, slow cell death, enable cell invasion, switch cell metabolism, or modulate immune responses. In the past 10 years, we've come to appreciate that these are often caused by upstream alteration in the splicing regulatory machinery, the splicing factor which regulate the outcome of the splicing reaction. Those can be mutated in tumors, they undergo alterations in copy number changes, transcriptional misregulation, for example, downstream of oncogenic transcription factors such as MYC, or changes in signaling pathways. Our lab and others are now working toward applying our understanding of the splicing mechanism and regulation to understand cancer pathogenesis and towards clinical translation. To achieve this, there are still a few roadblocks that we need to overcome. First, we need to better capture and quantify spliced isoforms. We need to integrate measurement of RNA dynamics and modifications. We need to measure tumor heterogeneity. And finally, we need to develop large-scale approaches for functional validation and clinical translation. Today's talk will highlight how we have been addressing two of these critical gaps. The first one on capturing and quantifying isoforms using long-read RNA sequencing, as well as applying our knowledge to understand novel clinical opportunities and predict drug responses. Once we have identified those splicing alterations in tumor, what can we do about them in the clinic, and why should we care about them? Well, beyond using them as biomarker, we can directly target splicing, and there are really two conceptual approaches. The first one illustrated on the top is by targeting the splicing machinery, for example, using small molecule inhibitors, and there are a number of those that are currently in preclinical or phase 1 clinical trials. The other approach is to target the tumor-specific isoforms. Those can be exploited for immuno-oncology, but can also be targeted directly to revert splicing towards the normal-like isoform profiles. And today's talk will highlight using splice-switching antisense oligonucleotide, which are small modified RNA molecules that can redirect splicing by blocking splicing regulatory element on the pre-mRNA molecule. Our lab overarching goal is to identify splicing alterations in cancer, age-related diseases, and rare diseases, decipher splicing regulation, and develop strategy to correct these mistakes. Over the past years, we have used RNA sequencing approaches to identify splicing vulnerability across a number of cancers, including breast and other MYC-dependent tumors, brain cancer, and gastric cancers. In addition to using short-read RNA sequencing, which are very popular, we have also leveraged long-read RNA sequencing, which is really a critical approach for capturing isoform diversity and complexity. While short-read RNA sequencing really takes apart one molecule and fragments it into thousands of small 100 nucleotide long fragments, long-read RNA sequencing enables us to see a molecule from tail to end. It is great to discover complex splicing events, full-length isoforms, a splicing of non-adjacent exons. And for this reason, it has really been a very powerful approach that has transformed not only genomics, but also transcriptomic. And we're now, us and others are now applying it to understanding disease-related transcriptomic changes. We have first applied it in collaboration with our colleagues here at JAX, the Beck, Paluca, and Bonshaw labs, to breast tumors. And by sequencing a large collection of primary breast tumor samples, we have identified thousands of novel isoforms that are not present in current reference transcriptomes. We show that half of those isoforms are really novel. A third of those are predicted to code for novel protein isoforms. And a subset of these isoforms are detected across other breast cancer cohort, including the Cancer Genome Atlas, and correlate with patient outcomes. We also show that a subset of those impact transposable elements. And so while this was really proof of principle of how complex the diversity of those isoforms is in tumor samples and how long-read RNA sequencing enables us to discover novel isoforms of clinical importance, we are now applying it to other tumor types and other diseases. And so in this talk today, building on this work, we have applied long-read RNA sequencing to a collection of lung tumors with the goal of discovering novel clinically relevant isoform. And this work was led by very talented MD PhD candidate Ryan Englander in collaboration with the Palooka and the Basho labs. And I will now let Ryan tell you more about his work.
All right. Thank you so much, everyone. Really excited to talk to you today about my work, and many thanks to Olga, my PhD mentor, and my co-mentor, Dr. Carolina Palooka, and the Bancho Lab, with whom we collaborated quite a bit on this project. So I'm really excited to talk to you about my work looking at using long-read RNA sequencing to comprehensively study the transcriptome of lung adenocarcinoma with a specific focus on a pair of intron retention events that we identified in the gene STAT2, which regulate the gene's function and predict response to immunotherapy. So without further ado, let's get into it. So before we talk about the data, I want to frame why we were interested in studying lung adenocarcinoma in the first place. As Olga had alluded to, we're doing this work in a variety of different tumor types, but lung adenocarcinoma in particular is one of the most common subtypes of cancer. About 75,000 cases a year are diagnosed in the United States, and it is one of the leading causes of cancer morbidity and mortality. The overall 5-year survival rate is merely 33%, and there are better therapeutic options now than there were just a few years ago, but they still remain limited for far too many patients. So the mainstays of treatment are surgery if tumors are caught early, but for most patients, they are diagnosed with locally advanced or metastatic disease. And for them, the mainstay of treatment is chemotherapy and radiation. And so those modalities can slow disease progression, but they rarely lead to durable cures. Targeted therapies like the EGFR inhibitors, KRAS inhibitors, and others have really come to the fore really within the past decade, and they've been effective at improving survival, as have the immunotherapies, which are a class of drugs that colloquially operate by taking the brakes off the immune system, if you will, and unleashing endogenous anti-tumor immune responses that can lead to durable regression of tumors. So when patients respond to these therapies, their responses can be quite deep and durable, but many patients either don't respond initially or relapse after an initial response and ultimately succumb to the disease. And so we sought out to ask the question of why that happens, with a specifically trying to understand how alternative splicing can contribute to the process of immunotherapy response and treatment resistance. And so to get a better understanding of the spliced isoforms that occur in lung adenocarcinoma, we turn to long-read RNA sequencing, specifically using the PacBio platform. So we took RNA from 12 lung adenocarcinoma patient tumors and conducted both long and short-read RNA sequencing. Long reads to identify novel spliced isoforms and full-length isoform structures, and short reads to provide important quality control and transcript validation. So our initial transcriptome was composed of, just from these 12 patient samples, of over 500,000 isoforms. We conducted quite stringent quality control filtering to remove artifacts and validate novel splice junctions, and this resulted in a lung adenocarcinoma transcriptome of over 180,000 high-quality isoforms, quite a high number, and we'll get into the breakdown of what that transcriptome looked like in a little bit. Just to give you a clue into what we did with our pipeline after that, we took our isoforms, we structurally classified them into several categories that reflect their similarity or novelty with respect to the reference transcriptome. And then we used a number of bioinformatic techniques, including some that we developed ourselves, to try to predict what the impact of each isoform was on the coding capacity of that gene. So we classified isoforms into encoding full-length matches to known protein sequences that have already been identified in the human reference proteome, novel sequences that are unique and would not otherwise be identified, and then truncated sequences that have a premature termination codon that leads to either a truncated or non-functional protein relative to the reference. And so zooming back out to that 30,000-foot view of what we identified with our long-read sequencing technology, we identified over 180,000 isoforms. To put that number into context, that is about 3/4 of the size of the human reference transcriptome at the time that we did this work. We did this in fall of 2022. So we identified over 75% as many isoforms as in the entire human reference transcriptome, which pulls from tissues from across the body. And so that took just 12 patient tumor samples in 1 month of a PhD student's time to assemble a transcriptome that large. Even more exciting, over half of the isoforms in lung tumors that we identified were structurally novel, meaning if we didn't do this sequencing, we wouldn't have been able to see these isoforms at all. And so what this indicated to us is that there are quite a number of isoforms in lung adenocarcinoma that currently are unannotated in contemporary reference transcriptomes. And so we're really excited about what modern iterations of this technology can do, given that it's improved dramatically just over the last several years since we initially did this work. So to bring it back to the initial question that we asked about how spliced isoforms might be impacting immunotherapy response and resistance, we took genes that were enriched for novel isoforms, having more novel isoforms than reference, and asked if there were any immune signaling pathways that were overrepresented among those novel isoform enriched genes. And we found that indeed there were 2 pathways that were enriched among genes with more novel than known isoforms. And these are the interferon alpha and interferon gamma response pathways that you see pictured here. And that was particularly exciting to us because these 2 pathways are known to be incredibly important in anti-tumor immune responses, both initiating them and in resistance. And so just by way of quick overview into what the interferon signaling pathways are, the interferons are a family of cytokines, most of which are in the interferon type 1 or type 3 families. And these are produced very rapidly after viral infection. They diffuse into neighboring cells after infection or ligate receptors on the infected cells and lead to activation of kinases that phosphorylate the crucial transcription factors STAT1 and STAT2. These molecules heterotrimerize with IRF9, go into the nucleus, and activate the transcription of hundreds of different genes that broadly lead to what is termed an antiviral state. And so this is a set of signaling pathways that's anti-proliferative, pro-apoptotic, and recruits adaptive immune cells and activates them in the milieu to kick off the adaptive arm of an immune response. And so these type 1 and type 3 interferons, which are also called the innate interferons, are essential for antiviral immunity, but also for anti-tumor immunity as well. The next group in this family, the type 2 interferons, there's only one that's been described. It's also known as interferon gamma. And so this cytokine is produced by activated T cells mostly, and it does broadly the same things or similar things, not exactly the same, but quite similar to what the type 1 and type 3 interferons do, but through a slightly different pathway that only relies on a homodimer of STAT1. So STAT2 and IRF9 are not associated with this process. But broadly, interferon gamma also leads to anti-proliferative and pro-apoptotic effects that have key roles in anti-tumor immunity. So we've known for quite some time that the interferons are important for anti-tumor immunity. They're essential for kicking off immune recognition, and they serve as incredibly important adjuvants that stimulate immune cells. And without them, anti-tumor immune responses are hard to organize and sustain. So in this sense, interferon is what we might call good. It helps initiate and sustain anti-tumor immune responses that can lead to durable responses in patients. But what we've recently come to understand is that interferon can also be bad in certain contexts. chronic interferon signaling inside tumors that occurs at low levels for a long period of time can lead to immunosuppression and immune exhaustion and actually contribute to immune evasion and treatment failure in immunotherapy and in anti-tumor immune responses more broadly. And so this dynamic balance between the anti-tumor and pro-tumorigenic effects of interferon is something that we're just starting to understand. And we were excited to look into our novel interferon-related isoforms and see if there might be some alternative splicing events and novel isoforms that might help shed light on the balance between the good and bad side of interferon in cancer. And so we functionally classified each of the novel isoforms in both interferon-related genes and across the entire long-read lung adenocarcinoma transcriptome and tried to look for differences between interferon-related novel isoforms and the rest of the novel isoforms we identified in tumors. And what we found, quite interestingly, was that interferon-related novel isoforms were enriched specifically in protein truncation events associated with a particular type of splicing event called intron retention. Now, that's particularly interesting because intron retention, unique among the splicing events, almost always leads to the introduction of premature termination codons that truncate protein sequences. Most splicing events, in order to predict something about what they do, you really have to know particular information about that splicing event sequence and its context, because introns are long stretches of nucleotides that frequently introduce premature termination codons. When they are included in a transcript, you virtually always know that the resultant protein sequence is either going to be truncated or non-functional. And so that was particularly interesting to us since these events were enriched among interferon-related genes, specifically in lung adenocarcinoma, suggesting that intron retention might be playing a role in allowing lung adenocarcinoma to evade interferon-related immune responses. And so to get a better sense of which specific events might be involved, we quantified alternative splicing events using short-read RNA sequencing from the Cancer Genome Atlas, comparing tumor to normal lung tissue with a specific focus on intron retention events in interferon-related genes. And what we found are differentially expressed events shown here on the slide. You'll notice that all of them are upregulated in lung adenocarcinoma relative to normal lung, suggesting that intron retention indeed might be a way that lung adenocarcinoma cells are evading interferon-related immune responses. I want to focus your attention here. There's really a number of interesting genes. We could talk about them for hours, but I want to focus your attention here on two intron retention events in the gene STAT2, namely intron 6 or RI6 and intron 12 or RI12. And that was is going to be the focus of the remainder of this talk. So by way of brief reminder, STAT2 is one of those three crucial transcription factors that allows cells to respond to type 1 and type 3 interferon. Without STAT2, you don't get responses to these cytokines at all. And so it is an essential component of interferon responses, both in cancer and more broadly. And so these are all of the full-length STAT2 isoforms that we identified with long-read RNA sequencing, segregated into intron-included isoforms that had either retention of intron 6 or intron 12, or isoforms where both introns are skipped. And so these isoforms on the top with retained introns are not capable of encoding the full-length functional STAT2 protein, whereas these isoforms below in blue theoretically are. And so to try to get a sense of whether or not these intron retention events could actually regulate interferon signaling in lung adenocarcinoma, we turned to a technology called antisense oligonucleotides, which Olga had alluded to earlier, to allow us to try to manipulate these events and study the impact of manipulating STAT2 intron retention on downstream interferon responses. And so antisense oligonucleotides are, as the name suggests, short oligonucleotides that are designed to bind specific target sequences in pre-mRNA. And they're chemically modified such that they do not induce transcript degradation when they bind those target sites. So they're in that sense, chemically inert. But what they do is they can sterically inhibit the association of specific regulatory RNA binding proteins at target sites and lead to changes in downstream splicing outcomes. So we designed ASOs that targeted either intron 6 or intron 12 and treated cells with those ASOs and interferon to study the impact of changing intron splicing on interferon responses. And so we took lung adenocarcinoma cells, A549 cell line, and treated them with ASOs targeting either RI6 or RI12, and looked at the retention of both RI6 and RI12 in those cells. It was actually particularly interesting that introns that targeted RI6 very frequently also had effects on RI12 and vice versa, indicating that there may be a coordinated splicing mechanism between these two introns. And so there's a lot of data here on this screen. So I want to focus in on two prototypical ASOs to really highlight the effect of intron retention in STAT2 and its effect on interferon signaling. And so if you look here at this first ASO, I want to focus on RI6 3'SS1. This ASO leads to a marked reduction in retained intron 6 retention, as well as a reduction in retained intron 12 retention here. That results in, at baseline, a trend toward an increase in STAT2 protein that becomes statistically significant when you treat the cells with interferon for either 6 or 24 hours, as you might expect. So more efficient splicing, removal of these introns leads to the production of full-length STAT2 protein that we can detect via Western blot. Alternatively, on the other hand, I want to focus in on this ASO RI12 3'SS1, which leads to an increase in retention of both intron 6 and intron 12. And that dramatically reduces STAT2 protein expression both at baseline and at 6 and 24 hours post interferon stimulation. And that actually has the expected effects that you might guess it would have on interferon signaling. So in the ASOs that increase STAT2 protein expression, namely RI6 3'SS1, we see an increase in the expression of 3 interferon-stimulated genes, MX1, OAS1, and RSAT2, at both 6 and 24 hours post-interferon stimulation. By contrast, the ASO that leads to a decrease in STAT2 protein expression, RI12 3'SS1, leads to a sustained reduction in interferon-stimulated gene expression at both 6 and 24 hours, indicating that ASO actually reduces the ability of lung adenocarcinoma cells to respond to interferon. So this leads us to the following model. STAT2 has these two introns, intron 6 and intron 12, that can be alternatively retained or spliced out in mature transcripts. ASOs can help us modulate that splicing either by inducing intron skipping that leads to productive STAT2 splicing and the production of full-length STAT2 protein that allows cells to respond to interferon and can actually boost interferon responsiveness after interferon treatment. Alternatively, some of our ASOs actually lead to improved or increased intron retention. This actually reduces the production of full-length STAT2 protein and concomitantly reduces STAT2 activity in response to interferon. And so this model was fairly straightforward and what you might expect based on what we knew about these intron retention events from the full-length data that we had from our long reads. But what we wanted to ask next was if these intron retention events that introduce premature termination codons that reduce the full-length protein, if they might actually themselves be capable of encoding protein that was just truncated, and hence might have different functions than the canonical STAT2 protein isoform. So these are the structures of the proteins that would be predicted from either complete STAT2 splicing or intron 6 or 12 retention. You can see the full-length STAT2 protein shown here with all of its known domains highlighted. This protein is fairly well described. There's 3 N-terminal domains, the NTD, coil domain and DNA binding domain that together allow STAT2 to interact with its binding partners and actually bind DNA. And then two domains here, the SH2 and transcriptional activation domains that allow it to be activated by phosphorylation and facilitate transcriptional upregulation when bound to target genes. So these moieties here are essential for STAT2's function in gene upregulation. You'll notice that both RI6 and RI12 lack those C-terminal functional domains. RI6 also lacks the predicted protein from RI6 also lacks the vast majority of the N-terminal domains that would allow retained intron 6 derived protein to bind its binding partners, but retained intron 12 actually retains most of those domains. And so we took these protein sequences and used AlphaFold to predict what their structures might look like. You can see the full-length STAT2 protein sequence here on the left with each of its different domains highlighted. STAT2 RI6 is a very short protein which has this extended piece of its coil domain that is somewhat disordered and hanging out at the C-terminus of the protein. Retained intron 12, by contrast, retains the globular nature of the N-terminal domains in full-length STAT2, but notably, as we mentioned, has lost those C-terminal transcriptional activation domains. So this raised the possibility to us that STAT2 retained intron 12, if it's produced as a protein, might actually be able to act as a dominant negative inhibitor if it retains the capacity to bind STAT2 binding partners, because it might be able to bind STAT1 and IRF9, but be incapable of actually transducing that transcriptional activation signal and hence lead to a further reduction in interferon responsiveness. So to test this possibility, we took A549 lung adenocarcinoma cells and created synthetic constructs, inducible synthetic constructs where we could express either an empty vector, retained intron 6 protein or retained intron 12 protein under the control of a doxycycline inducible promoter. We also included a flag tag at the N-terminus of both of these proteins so that we could detect these transgenes with Western blot. And you can see here on this Western blot that after treatment with doxycycline, RI6 and RI12 containing cells are detectable with an anti-flag antibody. We can also detect full-length STAT2 protein with a C-terminal antibody. So this is only detecting the wild-type protein induced from the endogenous locus, and the expression of that protein does not change. We also see, of course, nothing in the empty vector. And so when we look at the expression of two of our interferon-stimulated genes, MX1 and RSAD2, at various different interferon concentrations, you can see that neither the empty vector nor retained intron 6 protein lead to a reduction in interferon-stimulated gene expression at 6 hours post-stimulation, but retained intron 12 expressing cells do have a consistent across various interferon concentrations, consistent reduction in interferon-stimulated gene expression, indicating indeed that RI12 can act as a dominant negative inhibitor of the full-length STAT2 protein. So this leads us to this slightly more complex model for how introns 6 and 12 regulate STAT2 function in lung adenocarcinoma. So in the canonical case, productive splicing of both introns 6 and 12 facilitates the expression of full-length STAT2 protein, which is capable of transducing the response to interferon. Retention of intron 6 blocks the production of full-length STAT2 protein and either leads to non-functional or no protein production, and hence a relative inability for cells to respond to interferon. By contrast, retention of intron 12 alone can lead to the production of a truncated protein that can actually inhibit any remaining full-length STAT2 protein in lung adenocarcinoma cells, leading to a fully inhibited state rather than just a lack of interferon responsiveness. And so to get a better sense of how these introns might actually affect immune responses in cancer patients, which is, of course, our overarching goal, we turned to the clinical trial OAK, which is a large phase 3 clinical trial of the PD-1, PD-L1 inhibitor atezolizumab compared to the chemotherapeutic docetaxel. So we were able to access pre-treatment RNA from 503 lung adenocarcinoma patients in this trial who were treated with either the immunotherapy or the chemotherapeutic drug. And so we quantified splicing events in all of these patients, including introns 6 and 12, and looked for associations between STAT2 splicing and expression and patient survival. And what we found was quite striking. So this is a forest plot of hazard ratios from Cox multivariable survival analysis, which allows us to assess the independent predictiveness of each variable on patient survival, focusing on the immunotherapy treated cohort. So as you can see here, there are 3 variables that statistically significantly predict patient survival in response to PD-L1 treatment. That is, having a higher retained intron 6 is associated with significantly worse survival. Patients who have higher RI6, all else being equal, do worse than patients with lower RI6. By contrast, retained intron 12 actually has the opposite effect. It is associated with better survival in response to interferon in a statistically significant way. So all else being equal, more retained intron 12 actually predicts improved survival in response to immunotherapy. And the last variable that had a significant association with survival in this cohort was tumor mutational burden. Having a high tumor mutational burden was associated with better survival, and that's actually something that's been well described. Tumor mutational burden is one of the best biomarkers that we have. It's FDA approved as a biomarker for immunotherapy in a pan-cancer sense, but also specifically for lung adenocarcinoma as well. And so this survival picture was quite striking to us. It was a little bit surprising that retained intron 6 and 12 actually had different predicted effects on survival. And so we thought, well, if retained intron 12 is predicting better survival and retained intron 6 is predicting worse survival, maybe it's the difference between the two that is the strongest predictor of survival that we could have. And so for each patient, we calculated the difference between the splicing of intron 12 and intron 6. So we took the level of retention of intron 6 and subtracted it from 12, such that a higher level meant more intron 12 relative to 6. And we grouped patients into 2 cohorts, either delta PSI high or low. And you can see the results pictured here, and they are quite striking. About 15% of the patients in the atezolizumab arm of this trial were RI12 minus RI6 high, and those patients had markedly improved survival in response to interferon in response to atezolizumab, with a doubling in median overall survival relative to the patients in the RI12 minus RI6 low cohort. That is notably stronger effect than patients in the TMB high cohort relative to TMB low. There was a trend toward improved survival with these patients, but in this trial, it was statistically insignificant. And this is particularly notable because TMB, as I mentioned, is an FDA-approved biomarker in this patient cohort. So to get a sense of what transcriptomic changes might be occurring in these patients that may explain this notable impact on survival, we split our cohort into 2 groups: patients with intron 6 high and intron 6 low, and patients with intron 12 high and intron 12 low, and conducted differential gene expression analysis between each of these 2 groups to identify differentially expressed genes. We then took genes that were uniquely differentially expressed in either RI6 or RI12 and conducted pathway enrichment analysis on those unique genes to try to understand what the unique contribution of each intron was to the overall signaling milieu inside of these tumors pre-treatment. And what we found was that genes that were downregulated in retained intron 12 were uniquely associated with immune response pathways, in particular interferon gamma responses and interferon alpha responses. And so what this suggests to us is that higher levels of intron 12 relative to intron 6 are actually higher levels of intron 12 are preferentially associated with a reduction in interferon signaling, exactly as you'd expect, given that we know that intron 12 can encode an inhibitor of interferon signaling. And so that's quite a lot. So I want to draw us back 30,000-foot view and summarize what we found. So we conducted long-read RNA sequencing on lung adenocarcinoma patient samples and found almost 200,000 isoforms, over half of which were novel, with PacBio long-read sequencing. Really cannot emphasize enough, if we were not able to do this long-read sequencing and identify those full-length isoforms, we never would have been able to find what we ultimately did find about STAT2 signaling and responses. So this technology was essential for all of the downstream work that we've done. So in particular, we found two retained introns, intron 6 and intron 12, in the interferon response gene STAT2. When both of these introns are spliced out of transcripts, you have productive splicing that allows the production of full-length functional STAT2 protein that actually facilitates responsiveness to interferon. By contrast, retention of intron 6 leads to a reduction in full-length STAT2 protein and low levels of interferon signaling activity, whereas retention of intron 12 can lead to the production of an inhibitory truncated STAT2 protein that actually inhibits remaining full-length STAT2 and leads to a fully inhibited interferon signaling state. The first two groups of patients, those with high STAT2 activity and those with simply low STAT2 activity, do not respond well to interferon, but this last group, those that have this inhibitory protein, actually do show improved responsiveness to interferon, suggesting that it is potentially the ability of this inhibitory protein to inhibit chronic long-term immunosuppressive interferon signaling that potentiates later subsequent immunotherapy. And so the next frontier of our work, what we're really excited to do with the next round of long-read sequencing technology, is to move beyond bulk RNA sequencing. And so all the work that I showed you was done with bulk long-read sequencing, which of course does not give us any information about what cells express certain isoforms and where they are in a complex tumor microenvironment. And so with bulk sequencing, we lose a lot of really key information that we believe has tremendous insights for understanding the mechanisms of tumor initiation and progression, and also treatment response. And so we are incredibly excited to look at single cell long-read RNA sequencing and get full-length isoforms from single cells, and also work with spatial RNA sequencing to tell us where those full-length isoforms actually are in a tumor or in other disease states, to help us deconvolute what is happening in cancer and identify new targets for treatment therapy and mechanisms to overcome resistance. So with that, we'd really love to thank our collaborators across JAX and various different institutions that we have worked with. Would really like to thank our sources of funding, and of course PacBio for their incredible support and access to their amazing technology that's facilitated all of this work. And then of course all the members of the Anczukow lab, in particular Dr. Anczukow, who's been an incredible mentor to me and a role model. I'm so blessed to have come through the lab and have learned so much, and I'm really excited to have done this work and to move on to the next stage.